All right, welcome back to the deep dive. Today we're diving into something pretty big, a potential paradigm shift in how we think about cancer.
Well, absolutely.
You sent over this really interesting essay, The End of the Genetic Paradigm of Cancer from PLS Biology, just came out last month.
Hmm.
It really digs into some of the, well, cracks, I guess you could say, in the dominant theory, the somatic mutation theory.
Right.
The idea that cancer basically starts from genetic mutations in a single cell.
Yeah, that's been the kind of the dogma for a long time.
Yeah, for decades. Yeah.
Yeah.
So we want to really explore the challenges that are starting to emerge to this genetic view of cancer and also kind of look at what are the alternative perspectives,
Basically.
There's a lot of talk about organismal biology, cell state dynamics, tissue fields.
It's a whole different way of looking at things.
Yeah. So, I think the big question we want to start with is, are genetic mutations really the sole cause of cancer?
Right.
Or is there something more, is there a more complex interplay of factors?
Yeah, I mean, that's the million-dollar question, isn't it?
Yeah, that's what we're here to try to unpack a little bit.
Absolutely.
So, the somatic mutation theory, it's been around, as we said, for a while. It's really based on the idea that, like you were saying, cancer starts from a single cell that just starts behaving abnormally because of these acquired mutations in its genome.
Right, right. And, the advent of next-generation sequencing really, really pushed that theory forward.
Oh, yeah, for sure.
It gave this promise of like, oh, we'll be able to identify the specific genetic drivers, and then we could develop these targeted therapies.
Precision oncology.
Exactly, precision oncology. But, it hasn't quite panned out the way people thought it would.
Yeah, and that's what I thought was so interesting about this essay. It talks about there's this paradox. We've been able to sequence, so many tumors now.
We have tons of data.
We have mountains of data, and yet precision oncology, it's had some successes, but it hasn't had the breakthroughs that I think people were expecting.
It's not the magic bullet everyone was hoping for.
Not at all. Like relapses, they still happen.
All the time.
Yeah, and cancers, when they come back, can be even more aggressive.
And sometimes even harder to treat.
So it's like, this sequencing technology, in a way, it both strengthened the somatic mutation theory, but it's also revealed these fundamental inconsistencies.
Yeah, it's a double-edged sword in a way. We have all this information, but it's not always giving us the answers we want.
Right, right. And the essay even has this table where they list all these challenges, to the SMT.
Well, there are a lot of them.
Yeah. For example, like they talk about these tumors like rhabdomyosarcoma, epinymoma.
Yeah.
They don't have a lot of mutations.
They have very few or sometimes even no recurrent mutations.
Right. And yet they're still cancers. They're still behaving abnormally.
Yeah, and on the flip side, you have cancers that are caused by, known carcinogens.
Right.
Like chemicals. And the mutation signatures of those cancers, they can look exactly like sporadic cases.
Yeah, it's like where's the smoking gun?
Exactly. And then there's the fact that practically every gene in the genome has been linked to cancer at some point.
It makes you wonder, what does that even mean?
Right. If every gene can be involved, then is it really about the genes themselves, or is there something else going on?
Yeah, and then there's all this stuff about how normal tissues, they actually have mutations too.
Tons of them.
Including ones that are considered oncogenic, the ones that are supposed to cause cancer.
Right. But they're in normal tissue and everything's fine.
Yeah.
So how do we explain that?
It's one of those things that really makes you question the whole single cell, single mutation idea.
Exactly. And then you have like these precancerous lesions.
Right. Like moles or Barrett's esophagus.
Yeah, and they often have these driver mutations.
The ones that are supposed to be the big bad guys.
Right, but they rarely actually develop into cancer.
Yeah, and even when they do, it's often not even from the same clone. Right, like it suggests there's this field effect, like something's happening in the whole tissue environment.
Yeah, it's not just about that one cell that went rogue.
Right, and you even see this stuff with like radiation.
Like ionizing radiation.
Yeah, it doesn't immediately cause this like explosion of mutated cells.
No, it's more complex than that.
It's like multiclonal growth,
Yeah.
happens after several generations.
It's like something else has to happen first, something that's not directly related to the mutations.
Yeah. And then they talk about how, you'd think that if a cell had an oncogenic mutation, it would be like, selected for, it would have this advantage.
Right, survival of the fittest.
Yeah. But that's not always the case. Like they found that these mutations, they're not always under positive selection.
Sometimes it's just random chance, what they call neutral drift.
Right, right. And this is a huge problem for like the whole logic of precision oncology because it relies on the idea of there being this one target.
Right? You need that genetic fixation, like that one mutation that's driving everything.
But that's just not what we see in most tumors.
It's much messier than that. They
Yeah.
have all these different subclones, and they're evolving in these complex ways.
Yeah. And they even talk about how these early cancer cells, they can kind of hide out.
Like they go dormant,
Yeah, and then reappear later.
and that's what makes recurrence so tricky.
So it's like the whole picture, it's a lot more complicated than just a simple accumulation of mutations.
Yeah, the old story's starting to fall apart.
Yeah. And that's where this idea of non-genetic heterogeneity comes in, right?
Right, right. So single-cell transcriptomics, which is this really powerful tool that lets us look at gene expression at the individual cell level,
Yeah.
that's revealed that there's all this phenotypic heterogeneity even within cells that are genetically identical.
So like the same DNA, but they're behaving differently.
Exactly, and it's not just like some random noise. This variability actually has real biological consequences. Yeah. So you can have these cancer cells. They have the oncogenic mutations.
Right.
But their gene expression patterns, the way their genes are actually being used, it can look just like non-mutated cells.
Wow.
Yeah. And these states, they're not static either. They can change. They can transition between these different transcriptomic states. And that can be influenced by all sorts of things, internal factors, external signals.
So even if you have the mutation, it doesn't necessarily mean you're going to get the cancer.
Exactly.
Yeah.
And this is important for treatment, too, because it means that even within a tumor, you can have cells that are genetically the same,
Yeah.
but they're going to respond differently to drugs.
So it's like it's even harder to target them.
Exactly. You
Yeah.
can't just go after the mutation because the same mutation can be behaving in different ways in different cells.
So it's like the whole like one gene, one trait kind of idea.
He's out the window.
It's out the window, yeah.
The somatic mutation theory really relied on that, this idea that you could map a specific mutation to a specific outcome. But that's not how it works in reality.
So then what are some of the other things they talk about in the essay? Like they talk about these alternative isogenic cell states, right?
Right. So these are basically cells that can tolerate treatment.
Like drug resistance.
Yeah. But it's not because they have a specific mutation that makes them resistant.
Oh, okay.
It's because they're in a particular state, a persister state. And these states, they can actually be reversible.
So it's not like a permanent change.
Exactly. And that's another challenge to the somatic mutation theory, because it assumed that resistance was always due to these acquired genetic changes.
Right, that were permanent.
Yeah. But with persister cells, it's like they're just kind of hunkering down, waiting out the storm.
Interesting. So how do they get into these persister states?
It can happen spontaneously, or it can be triggered by signals, like from the environment.
So the treatment itself could actually be inducing resistance.
Exactly. It's like this unintended consequence.
Wow. That's kind of mind-blowing.
Yeah, it really changes how we think about treatment. And then there's this whole thing about stem-like cells.
The cancer stem cells.
Right. So for a long time, people thought that these were just kind of pre-existing, like you had this population of cancer cells that were already stem-like.
Okay.
But now it's looking like these cells can actually arise in response to stress.
So like the treatment is making more of them.
Exactly. It's like an active response, not just passive selection.
Wow. That's a whole different way of thinking about it.
Yeah, it means that we can't just focus on killing the existing cancer stem cells. We have to think about how to prevent new ones from forming.
So it's like we're not just fighting the tumor, we're fighting the environment.
Absolutely. It's a whole ecosystem.
So it seems like there's a lot of evidence now that this simple story of cancer being driven just by gene mutations, it's not holding up.
It's getting harder and harder to ignore the cracks.
Yeah. So where do we go from here? What are some of the alternative ideas that they talk about in this essay?
Well, they spent a lot of time discussing these ideas of like phenotypic plasticity and tissue organization.
Okay, let's break that down.
Sure. So phenotypic plasticity, it's basically the ability of cells to change their phenotype, their behavior,
Yep.
without any changes to their DNA.
So like they're adapting to their environment.
Exactly. And this can be a really important factor in cancer development because it means that even if you have a cell with a mutation, it doesn't necessarily mean it's going to become cancerous.
Right, it depends on the context.
Exactly. And then tissue organization. It's all about how cells are arranged within a tissue and how they interact with each other and with their surroundings.
So like the structure of the tissue matters.
It matters a lot. It
Yeah.
creates this whole ecosystem, this environment that can either promote or suppress cancer development.
Interesting.
Yeah. And one of the key things they talk about in the essay is the tumor microenvironment.
The stroma.
Right, the stroma. So this is all the stuff that surrounds the tumor cells, the blood vessels, the immune cells, the extracellular matrix. And for a long time, people thought that the stroma was just kind of passive, like it was just there to support the tumor.
Like a scaffold.
Yeah, but now we're realizing that it's actually playing a much more active role.
So it can actually influence the tumor.
Absolutely. It can send signals to the tumor cells. It can provide nutrients. It can even help the tumor evade the immune system.
So it's like the stroma is almost like a co-conspirator.
Exactly. It's not just an innocent bystander.
Wow. So it seems like we really need to shift our thinking from just focusing on the tumor cells themselves to looking at the whole tissue environment.
Absolutely. It's not just about the seed. It's about the soil.
That's a great analogy. So if we're starting to move away from this purely genetic view of cancer, what does that mean for treatment?
Well, it means we need to start thinking about new approaches. Instead of just trying to kill cancer cells, we need to think about how to reprogram them, how to get them to behave normally again.
So like re-educating the cells.
Exactly. And we need to think about how to target the tumor microenvironment, how to make it less hospitable to cancer.
So it's like we're not just fighting a war, we're trying to create peace.
That's a good way to put it.
So what are some of the specific things that we could do?
Well, there's a lot of research going on right now into things like immunotherapy, which harnesses the power of the immune system to fight cancer.
Okay.
And there's also a lot of interest in developing drugs that target the tumor microenvironment.
Interesting.
Yeah, and then there's these more holistic approaches, like lifestyle changes, diet, exercise, stress reduction.
So it's like taking a more integrative approach.
Exactly. It's not just about finding the magic bullet. It's about creating the right conditions for the body to heal itself.
That makes a lot of sense. So it seems like we're really at a turning point in cancer research.
I think so.
Mm.
We're starting to realize that the old paradigm is no longer sufficient.
We need a news story.
We do. And this essay, it's a good start. It's challenging us to think differently about cancer, to look beyond the genes and to see the bigger picture.
And to realize that the body is not just a machine, it's a complex ecosystem.
Absolutely. And if we want to truly conquer cancer, we need to understand that ecosystem.
I couldn't agree more. Well, this has been a fascinating conversation. Thank you so much for sharing your insights.
It's been my pleasure.
And to our listeners, we hope this is giving you some food for thought. The future of cancer research is wide open, and we're excited to see what new discoveries lie ahead.
Me too. It's an exciting time to be in this field.
Absolutely. Until next time, keep diving deep.
And keep questioning everything.
Exactly.
There's always more to learn.